Hi, everyone. Thank you very much for joining this morning, afternoon, evening, wherever you are in the world. Middle of the night, maybe. I don't know, Lisa, from where you're joining, but I'm very happy that you're all here for Julia Kurz's talk. She's from protocols.io, and she will present today. You probably can already see her screen, a tool for core facilities to share their methods. And we're very happy to have you here, Julia. We have a full half hour for discussions, and anybody, whenever you have a question, you can paste them in the chat or raise your hand, and I will call you then. So yeah, I would say without further ado, Julia, you can start. Thank you very much for joining us.
Thanks, Sophie, for having me, and good morning to everyone who's joining, or good afternoon, wherever you're located. So as Sophie already said, I'm Julia. So I'm a scientific engagement manager at protocols.io, which is a platform for documenting and sharing research methods. So I started off as a researcher. I did my PhD in chemistry. And when I look back, there were so many different facility staff that were really instrumental in me completing my PhD and getting the data that I did. I'll just shout out my NMR spectroscopy technician, Tree, who was super helpful during my time. But it's really nice that now part of my role is working with facilities and showing them how protocols.io can be used to enhance the credibility, visibility, and efficiency of those facilities. So you'll see on these slides here some examples of researchers expressing their frustration at the lack of information in the published literature for repeating experiments. And so a little bit of history, this is why protocols.io was originally started. So it began as a startup company in California about 10 years ago to solve the problem of reproducibility in research, a lack of people sharing detailed methods, and kind of lack of opportunities for people to get credit for working really hard on developing those methods. And so fast forward to now, protocols.io is part of Springer Nature, and it's coming more and more integrated into different research and publishing workflows. So it has evolved over the years, but at its core, its purpose is to make it easy to share method details before, during, and after publication. So there are two sides to the platform. There's the public side where we act as an open access repository for publishing and discovering scientific methods. So when the protocols are published on the platform, they receive a DOI so they can be cited. There's also the private side of the platform, and that is a secure workspaces where teams can create and collaborate on methods before they're published or if they don't choose to publish them at all. And really, these two sides of the platform kind of feed into each other. So for example, the published protocols on the platform can be really easily repurposed and brought into the private side of the platform. So you can create your own private editable copies of these published protocols via what we call versions or forks, so that you can adapt them for your own specific experiment or laboratory setup. And what this means is that you can track the reuse of the protocols that are published on the platform, seeing how many people are forking those protocols for use in their labs. And then when teams really utilize the private side of the platform as a workflow tool to document and keep track of their methods, it then becomes really easy for them to push those protocols public. So it kind of reduces the burden associated with writing up a method and then sharing it publicly because it's already documented on the platform. You just need to click a button to publish it. And so this slide here is really to kind of demonstrate that when I say protocol, I'm not necessarily talking about kind of a PDF document that you upload and then it receives a DOI, but the protocols on the platform are highly dynamic and interactive. So you can see a little bit what they look like there. But what you can do is you collaborate with your colleagues in real time. I've already kind of touched on the versioning and forking that's in place to ensure that the changes to those methods can be tracked over time. You can add various things, what we call components, videos, images, to make sure that those protocols are very user-friendly and understandable to people outside of your lab. And then all of those protocols, when they are stored in shared workspaces. So it's easy to find what other people are working on in your department or in the broader research community. When you do keep your protocols on the private side of the platform, they are secure, and you can kind of configure the settings how you want to do things like, for example, who can just view the protocols, who can edit, who can publish, things like that. And then some other functionality that I wanted to highlight today that goes alongside the protocols are what we call run records. And this is where you can turn your protocol into a checklist that can be followed in the lab as an experiment is being completed. So if we think about the protocols themselves, that's what steps should be taken. But we know that, things happen in the day when an experiment's being done that mean that you might have to change something. So the run record can then be used to capture the day-to-day variability in how those protocols are actually being followed. And so you can record things like metadata, comments about how a specific step went, any observations, alterations, data points. So you know how an experiment was run, and then you can correlate exactly the method that was followed on a specific day with the data that came out of that experiment. You can kind of put these run records alongside your data in various places. You can export them to an electronic lab notebook or your other kind of cloud-based drives to really have them sitting alongside the data. And so the kind of dynamic, interactive format of the protocols means that the methods on the platform follow FAIR principles. So if anyone isn't already familiar, you can kind of see a little bit of an explanation there on the slide. So it kind of originally, FAIR has been a conversation around data. Data should be findable, accessible, interoperable, and reusable. But if you think about sharing data, if you're not sharing the methods that were used to generate that data, can people kind of truly understand the data, reproduce it? And so that's why it's really important for methods to be fair, as well as data. And so the methods on protocols.io have been kind of specifically designed to comply with FAIR principles. So you can see it there kind of along the bottom. They're findable, they're publicly indexed, and even on the private side of the platform, it's very easy to kind of search for and find the protocols that you're looking for. They're accessible. All the published protocols are open access, and they have a CC BY license. We also have an open API, so you can kind of build up integrations that you want. And we also have existing integrations with electronic lab notebooks and cloud storage platforms to ensure that those methods are interoperable kind of across different areas of the technology landscape. And then the built-in versioning, forking, and running functionality on protocols.io ensures that those protocols are really easy to reuse. And also that the provenances or kind of, paper trail of credit is maintained back to the original protocol developers, even if those protocols are kind of changed and adapted over time by different authors. This might be something you've kind of already heard of, but essentially the kind of publishing industry is moving away from the more, I would say, historical way of publishing a single research paper into what we call modular publishing. And so this is where the paper might or might not detail the kind of story or narrative around a piece of research work, but all of the other outputs or modules associated with that work are shared in fit-for-purpose repositories. So that could be things like molecular structures, code, images, data, and of course, methods and protocols. So on the right of this slide, you'll see a nice example of what that kind of looks like in practice. So this is a Nature Communications paper, and we can see the data availability section of that paper. And they've said that the expression constructs are deposited at AdGene, the protocols on protocols.io, and the molecular structure in the protein data bank. And so this is an example kind of from traditional research, but what this could mean for a facility is that the technician involved with developing a method can then take ownership of that research output that is distinct from the research paper. So this is a deeper look. That's the same paper that I showed on the previous slide. So that's the paper at the top. And then underneath that is the protocols.io protocol that was linked within that paper. And so we can see that because these are separate research outputs, they can have separate author lists. So in this example, we can see Kelsey Ramirez. She was a researcher. She was fourth author on the paper, but because she was the primary protocol developer, she could be first author on that protocol. And so I think, we can acknowledge that facility researchers, they're not always going to be authors on papers, but you can be the author on a protocol, and it allows you to kind of take some of that control back, publishing your protocols on the platform, and then the researchers you work with can cite them, or other people can find them and reuse them as well. In addition to credit, kind of another relevant topic that ties into the FAIR methods is the accessibility of those methods. So you might not always have control whether a method that's detailed in a traditional paper, if that paper is published open access or via subscription-based model. But if you're putting your methods on protocols.io, they will always be open, even if kind of the paper that's linked to those protocols is published in a subscription-based journal. So we can really kind of have the transparency of those methods across all the different publishing models. So when methods are linked to a paper, we know that the kind of science doesn't stop after publication. So on protocols.io, the published protocols can be updated over time. So as you make your own internal improvements to those methods, you can share them kind of with the broader community. So we see here, this is version 1 of a protocol on protocols.io, and that was referred to in this PLOS Biology paper. But if we go and have a look on this next slide, so if someone actually comes across this paper and follows this DOI link, what they will land on is a page that looks like this. And so we can see they are landing on version 1 of the protocol, which was the version that was published at the same time that this paper is published. But they're notified that there is a newer version of that collection of protocols that's available on the platform. And so they can always be aware and working from the most current and up-to-date method that's in the literature. And so this is a kind of another example of what that looks like in practice. So again, in this example, it was from researchers and they were involved in publishing the protocols and the paper. But you can see that version 1 of that protocol was published at the same time they published a preprint. They then were able to update that protocol on protocols.io right before they published kind of the journal article. And then even after that journal article was published, they updated the protocol again, nearly over a year later. And then because it's on protocols.io, all into the future, that protocol can continue to be updated and built upon by the authors or by other people in the research community via forks. And so what this might mean for a facility is that you might have kind of a standard protocols that are used in your facility, but that protocol might kind of drift and change over time. So you can capture and publish all of those versions over time. And then all the researchers that you work with, they might be also kind of concurrently publishing papers that are associated with those different protocols, version 1 or version 3. And they can all be kind of linked together on the platform, and we can see how they're related. So let's kind of bring that back to facilities a little bit more. So, I think probably everyone in the audience can agree that, facilities are really central in expediting research. But, what we hear is that it is a struggle to kind of demonstrate impact and receive recognition. So hopefully from what I've shown so far, you're kind of already thinking and having an idea of how Protocol.io might fit in and help. But let's kind of talk about that together now. So if we think about the research ecosystem, the facility would provide, equipment, expertise, and really hands-on support that kind of makes good science possible. That knowledge and experience will feed into methods and protocols, which are being, tested by the facility in real research contexts, being refined by the facility, and kept up to date. Now, that can happen in a variety of ways. It might happen on paper, on kind of random notes, through conversations, or just information that's stored in people's heads. But at the end of the day, those protocols really can empower researchers to generate data, ultimately publish papers, and kind of generate the impact that is associated with that research. But how can we ensure that that impact and recognition comes back to the facility? So what you can do is, as a facility, you can maintain a library of protocols on protocols.io. This means that instead of being kind of in people's heads or in random notes, those methods are in a central location. They can be published, they can receive a DOI, so they're citable, discoverable, and connected to the wider research community. Then what the researchers can do is they can easily find and see kind of the latest version of protocols that the facility is working on. They can follow them if appropriate, and then they can cite them in papers. We understand that kind of in practice, not every citation or attribution happens perfectly, but having your protocols on protocols.io, it means that your work is kind of visible more broadly and not kind of dependent on the paper. There are also things that you can do on the platform. So if you find out that there was a paper published that's associated with your protocol but doesn't cite your protocol, you can kind of add it into your protocol after the fact to show, hey, we published this protocol on protocols.io, but it's actually feeds into, for example, this Nature paper that was published. But yeah, as I kind of mentioned earlier, if they're on protocols.io, other researchers can find them and use them, which kind of naturally expands the facility's reach and impact. And also when they're on protocols.io, they will have their own independent impact metrics. So this can be things like citations, forks on the platform, and those are all visible on the individual protocol level, and also kind of in an amalgamation of all of the work that might come out of the facility. So what this would look like is something like this. So these are some workspaces on protocols.io. And we can see there's kind of multiple tabs along the top here. But if we look at this impact tab, we can see kind of all of the different publications that have published the protocols within the workspace. And then we can see at the top here a kind of group of the total number of citations and views for all of the protocols published as part of that workspace. So this workspace on the top here, it's from Quark-Blimy, which is an initiative focused on improving the kind of quality and reproducibility of data generated from light microscopy. And then this is another different workspace for the Human Cell Atlas community. And we can see here, so this one's a little bit more popular. It's got many protocols, lots of different members of that workspace. And we can see that there's a lot of impact that's being generated by that workspace. But what's really important as well, I think it's not just the facility that can get credit, but also the staff within that facility. So you can have a workspace on protocols.io for the entire facility, but there's also the ability for individual researchers to have their profiles on protocols.io so that they can kind of demonstrate, hey, these are all of the protocols that I've developed, and that might be relevant if they kind of go into conversations about kind of career development, but it also just allows them to demonstrate their expertise in a specific technique. What I'm going to quickly do now is run through a couple of examples so you can see what people have done on the platform. So what's shown here is this is a collection of protocols. So that's actually published by the same researcher that I showed on the previous slide. So Ronan, he's the author here. This collection of protocols, it sits within the facility workspace. So this is the facility. It's part of the University of Manchester. And so he's published a collection of protocols on how researchers can do the sample preparation for using that facility. So the researchers can find that and follow those protocols. This is another kind of imaging focused protocol. And what I want to show here is that the information that can be put in that protocol. So you can see that it is a step-by-step instructional process and that there's various information that can be added. So you can add in timers they work as actual timers. You can detail how long each steps in that process will take. And then there's various things to show that this is going to be an incubation. You might need to do pipetting here. But really, this is kind of scratching the surface of everything that's possible with the platform, which I unfortunately don't have time to show everything today. But please feel free to take a photo of this slide. These are some of the example protocols that I showed in the previous slides. And I think it's really nice to be able to go onto the platform, play around with those protocols and see exactly what's been done. That's really the best way to see what's possible. So yeah, please, I encourage you to check out the platform, check out these core facility and kind of imaging related protocols to see how you might be able to kind of do that in your own work. So before I kind of finish up, so these are the many organizations that have recognized the need for a better way to capture and share method details. What I really want to highlight here on this slide actually is the funders. So many funders now recommend and sometimes even mandate that if they're funding a piece of work, that those methods should be shared on protocols.io. So one of the reasons for that is that it gives those funders a clear way to demonstrate and track the impact of their investments. So when you as a facility are sharing your methods on protocols.io, you can really demonstrate the value and return on the investment that comes from any funding that is used to fund your facility. And then this is my last slide. I wanted to let you know about another webinar that will be running later this month. So if you want to hear from kind of your peers instead of from me, what we'll be doing is hosting 3 core facility leaders. And I think so Dr. Britta and Dr. Ralph, they're from imaging facilities as well. And they'll be talking about the role of core facilities more generally and the kind of role that protocols.io plays in that. If you're not sure if you can make it, you can always just register and you'll receive the recording afterwards as well. Yeah, thank you everyone for listening. Please check out protocols.io. That's the URL. It's just protocols.io. And I'm happy to answer any questions.